I think everyone is exhausted, and I'll give a spicy bite, very short and spicy bite, last punch. So before going to first. So this is actually a team led by Dr. Madhavi Reddy madam. We are like a seeds attached to the placenta, and as you know, chili, the pungency comes only in placenta, and entire ideas were from the Dr. Madhavi Reddy madam, and we are like seeds just worked on these aspects. So this I don't want to touch. You all know from the morning onward they are different, but there is a huge declining trends. It's not going to be that spicy in future, and we are going to be not in the first position. Our position may drop if it doesn't work together. So only four traits I covered today: Begoma viruses, Anthraquinose fruit rot, and Southeast Asian thrips, and yields we required RF and cytoplasmic meal sterility. So first trait, Begoma virus resistance for chili leaf curl virus. 3 resistant sources have been identified. Dr. Madhavi Reddy madam and Krishna Reddy sir. These 3 were extensively used in F1 hybrid development, and madam has developed very good hybrids, which are popular in Karnataka, Andhra and Telangana also. We tested. As we know, already sir pointed out, based on the strain and based on the environment, it may vary. And then recently we identified another 4 sources, mainly in capsicum chinense and capsicum fruitescens. These are also having good resistance to the chili leaf curl virus. So these are the hybrids developed for the chili leaf curl virus by Madhavi madam. These are the new hybrids, Nihira and Dhriti. These combined tolerance to phytophthora and leaf curl resistance has been developed. So mapping regions, we went for BSA QTL-seq approach for the mapping the genomic regions, and we could identify in the chromosome 6. And we developed the 2 SSRs marker through targeted locus-specific development of markers. So we identified the region first, and then the targeted locus-specific markers have been developed, and 2 markers have been developed through BSA QTL-seq approach. And you know, Punjab, chromosome 6, there was only 1 report from the Punjab. That resistance also has one chromosome 6. So, but that marker is 40 centimorgan away from our gene. It clearly shows that both are different genes. Punjab source and our source, both are different here. So we validated that in, and in another alternate population also, it's working very good, around 89 to 90% prediction efficiency. You know, in chili, it's very difficult to land very precisely because of the huge genome size. It's very difficult to get 99% or near to the fine mapped position. It's not working. So these are the publicly available sources so far. PEPI1, PEPI2, already we know, very pioneer, that PEPI1, PEPI2 are not working in our sources. Those genes are not there in our sources. And PEPI3, we thought Punjab has reported earlier to us, it might be the PEPI3 gene. We have reported on chromosome 6 another source, that is maybe PEPI4. And another, even private companies might be having so many resistant sources, it might be so many PEPI genes might be there. So this is what we require to combine all the genes for the durable resistance. Okay. So this we have checked for PEPI genes also, it's not working. So we don't have PEPI genes in our sources. So recently identified sources in capsicum, chinense, fruitescens, we studied the genetics. So earlier identified sources for single dominant gene action. This was single recessive gene action, both are allergic in nature. We have found one in capsicum chinense, another in capsicum fruitescens. And then when we went for BSA QTL-seq approach, again we landed on different chromosome. It's on chromosome 5 and 11. So same virus, but we landed on chromosome 5 and 11. So this might be another PEPI gene conferring for the resistance. It clearly indicates that there are different genes acting for the different viruses are different based on the source also. So if you want BrahMos for leaf curl virus, you should breed pyramid all the genes into the one background. That is the only option available for the Begoma virus resistance. So entire work in the short form, 3 good sources have been identified, which is single dominant gene action. And then we identified markers on chromosome 6 for chili leaf curl virus. New sources identified in the capsicum chinense and fruitescens. These are single recessive gene, which are on again chromosome 5 and 11. So this is what we are doing. We further now our next target is to combine all the genes into the one background. This is about chili leaf curl virus. Next comes to most important next target rate is anthracnose fruit resistance. It's been 30 years over. PVC 80, 81 are the only sources available for the anthracnose fruit resistance. Under NASA project of Dr. Madhavi Madam, this work started in 2016-17. We studied the genetics and its inhibitor gene action, 13:3 ratio, inhibitor gene action. Then the population. Then population, we went for the GBS, genotyping by sequencing. Through SNP markers, this is a linkage group of that population, F2 population. We identified regions on chromosome 2 and 6. Two regions have been identified having conferring resistant to the anthracnose fruit resistance. So this again, our hypothesis has been proven, 13:3 ratio, two genes, and here also we got two regions exclusively on chromosome 2 and 6. So this was a very huge region, 15.12 MB on chromosome 2 and almost 5.09 MB on chromosome 6. So this we fine map through target locus specific markers, which has been targeted, and the fine mapping position now it's chromosome 2, it's 836 kb, and chromosome 6 is 139 kb, and as already we mentioned genetics, these two are epistatic in nature. And with this markers on the both chromosome 2 and 6, if you use combinedly in the epistatic gene action, almost 74 to 78% we can predict. This is good in case of chili. So these are the different selected genes in chromosome 2 and 6. As in refined positions, there are only 12 genes which can be used for the further validation. So this is again, I want to point out why other sources, other markers doesn't work. You can see we have landed only in chromosome 2 and 6 for our strain. No reports are there so far on this chromosome. So all the other countries, Taiwan, Thailand, wherever they work, they have different genomic regions identified. So this clearly shows that our strains are different. We need to work for Indian strains only. So next approach, we went for the incorporation of resistance into the Annam background. We tried Annam into Bacchatum, we failed. There is a huge embryo abortion. 1-2 plants we could get, but it also set fruit, but again, the embryo abortion was there. It's failed. We tried with, sorry, this is not working. You can move at least. Excuse me, you can only move. It's not working. Okay, sorry. So Chinans also we tried, and here also same problem. We could not succeed the hybrid, and this is the hybrid weakness. The plant stayed for more than 1 year but doesn't flower. So this is the hybrid weakness, and whatever fruit set, there was no seed inside. So normally we think this is a virus, but it's not the virus. It's a growing control condition, but it's a hybrid weakness. So with the fruit sets also we tried, we also failed. Same thing, hybrid weakness, no fruit set. Even it set also very aborted fruit. So we went for the bridge cross strategy, capsicum annum into Chinans, and then the Bacchatum. We could develop the population through backcross strategy. So we developed 3-way hybrid population, then went for the backcross progeny. This is how in the 3-way hybrid backcross progeny, still there was seed set was the problem. Then we backcrossed, now we are self-fruit net started. In the backcross generation, this is the problem. Pollen viability is a big issue. It was not setting through its own pollen. So we again backcrossed with annum. Now the plants are ready, and the families are ready with the self-fruitness. Then the families we selected through marker cluster selection. Those families we screened for the anthracnose fruit resistance through in vitro method. We have found very good variability. We are having good leads. Good families are there which are showing good tolerance in the in vitro. So both the gliosporids and truncatum we tried, and we got good families with introgressive loci. So far this is the stage we are there, and this population has been forwarded expecting good results in the next generation, because we are in BC3 stage. Breeding for anthracnose in a nutshell, 2 QTL regions on chromosome 2 and 6 we identified. We fine mapped that into KBs, and now the markers are available to predict the anthracnose resistance, and we introgressed through a bridge cross strategy, and we have good families having tolerance to the anthracnose fruit resistance. This is the nutshell for the anthracnose fruit resistance. Next, breeding for Southeast Asian thrips. I think no one talked from the morning. Southeast Asian thrips is a big problem. We tried for first standardization of protocol. We have standardized the scoring method And this is the scoring method on flower, leaves and fruits. So 0 to 7, we have standardized the scoring, because scoring itself is, if it goes wrong, the screening will be difficult. So that's why we standardized. So we screened, this is a scoring, sorry. Okay, we screened extensively 481 accessions in the field screening based on the score. Then we went to selectioning the accession 60 accessions, 34 accessions in the polyhouse condition. Then we moved into whole plant bias, 16 accessions. Then we narrowed down to 4 accessions. Now we have 4 accessions which are showing good tolerance to the Southeast Asian thrips tolerance. And through multi-choice and no-choice assay, we have identified good accession, IHR BHP79, which is fruit sense resistance. So this is a nutshell. All the data is there here showing this one. But in a nutshell, we identify the resistant source in the capsicum fruit sense, that is IHR BHP79 having good tolerance to the Southeast Asian thrips. But if you given a no choice, it will eat away. So if it is under multi-choice assay, it's holding the good tolerance. Next. This is how we have done in the in vitro screening. We have scraping percentage also we calculated based on the ImageJ software. Next, please. So this is a source available for the Southeast Asian thrips. Next. This thrips tolerance has been found to be the antibiosis, because it has some metabolites based on the thrips is not developing its own life cycle. Next, please. Then we identified metabolites in the resistant source through GCMS method, and we identified good metabolites for the thrips resistance. Next. So there are 3 repellents in the resistant source which are are in the resistant source. Next. So these repellents have been tested through synthetic chemical also. It's showing good repellents action against the thrips. Next. We studied the genetics, and as we expected, it's polygenic recessive. It will be very difficult to breed thrips resistance as it's more polygenic recessive in nature. Next. We tried markers also. We have screened a lot of markers, around 800 markers. And as you know, till now only Franklin accidentalis markers are there. And it's a patented technology in USA. Those markers we tried and it's not working. It shows that Franklin accidentalis markers, regions, and our thrips parvispinus regions are totally different. Next. So we tried with the 748 markers, we got only 0.67% low polymorphism. This is another bottleneck in chili breeding. We will not get polymorphism, very less. It will be in single digits always, maximum to the 10 to 12%. Next. In a nutshell, we standardized the protocol for thrips parvispinus resistance. We identified the source having good tolerance in the capsicum fruit sense. We identified the repellent compounds in the source, which can be used as go for the IPM technology also as a repellent strategy. And resistance is polygenic recessive nature, and we identified 2 markers showing very less PVE, 8.63%. Next, in future, we are trying going for the next generation sequencing protocols to develop the markers for the thrips resistance. So this is about Southeast Asian thrips. Next. Very important for the hybrid development, we need sterile cytoplasm markers and also RF markers. And sterile cytoplasm marker, many markers are available, ORF or 456, ATP6-2, and recently marker 16 and 18 from Joe et al. Also we tried. Only caution I want to give is that based on the concentration, the marker errors will come. If you see the 20 nanogram per microliter DNA, there is a difference, and 30 nanogram light banding is coming, and 40 nanogram per ml it's coming different. So this is due to substoichiometric effect. We have to be very careful when using sterile cytoplasmic markers. Fix the DNA concentration and use across same. If you change the concentration of DNA, there will be errors in your scoring. Next. So RF marker is very important if you want to change the background into different backgrounds, CMS backgrounds. For RF markers, we went for genotyping by sequencing GBase in F2. We landed on again chromosome 6. That SNP we converted into DCAPS marker. That is the amplification profile of DCAPS marker, which is around 7.9 centimorgan away from the RF gene, and which will be very much useful for the changing the background and also in hybrids testing. Next. And again, I want to tell that there are 2 reports on RF markers till now. Ours will be 3rd report in the public domain. I'm not telling about the private, in public domain. So those 2 markers also not working in our RF gene. So it clearly indicates that the markers in chili are mostly genotypic in nature. you cannot use whatever, published marker without validation. So most of the markers are genotypic specific in nature. This is again the third bottleneck in chili breeding. Next. So this is overall about our work. Constraints what I observed is crop and climate. So climate is a big issue now. Growing crop itself is becoming difficult. We are getting short duration for the breeding program. Because if you delay the planting, the temperature is rising and we will not get chaffwood seeds. Second is often cross-pollinated more than 65%. If you miss chance in selfing, you will lose the generation and you will go back. Third is larger genome size. As I said, very difficult to pinpoint the genomic regions. It will be very difficult. You cannot find like tomato and all. It cannot be possible in chili. Third is markers are highly genotypic in nature. So it cannot be reproducible across the genotypes. Very tough in chili. And fifth one, everyone is speaking, the collective collaboration efforts really not there in chili because we need to come for the gene pyramiding approaches, different sources approaches. This has to be done. And varied states in pathogens. Many of the sources, many of the markers reported elsewhere in Taiwan, Thailand doesn't work for India. So if you want to breed for Indian source, we only have to collaborate and cooperate and work. This is because based on our work I am telling. And next, varied strains and wild species no one touched so far. So always many speaks that wild species means capsicum, China and fruit sense. No, they are all domesticated species. There are 5 domesticated species. Wild species like capsicum toveri, capsicum pubescens, capsicum galapagos still not been explored, not been tried. So that also another area we need to think, exploring those genotype, those accessions for the breeding program. So with this, thank you very much. Thank you.